Okay, let's go through 29 and what I want you to really know. So when we look at 29, the first few slides and the first probably half of it is all about how fertilization occurs and then what happens during the process after fertilization. Now, big picture stuff. Big picture stuff is somehow out of one cell, a cell that's a fusion of one sperm cell and one oocyte, they combine to form only one cell. Somehow we have to make hundreds, probably 100 trillion cells out of it. So you can imagine how complex it is, right? Out of one cell, you got to form trillions and trillions of cells. And you can't add more cells from the outside in. So what we need to do is kind of, have cell division, form two cells, those two cells grow, and then they divide again, and then you have cell division again, and then you're going to grow, and then you're going to divide and have mitosis and completely split again. You can imagine how long it takes before you go from one cell to even 1,000 cells, right? Now, it's going to take more than 1,000 cells to become even a small fetus. So it's going to take a long time with multiple rounds of DNA replication, followed up by division, right? By mitosis. Now, with the first part right here, in this slide, fertilization right here, take a look at what happens with fertilization. When the oocyte is released, that released oocyte has cells surrounding it that are going to help protect that oocyte, that even after ovulation, those cells are present and help basically defend the oocyte. Now, what else do we see are that you have a gel-like layer called the corona radiata, then that's made out of the cells. And if you take a look really close, you have a real thin, even more substantive gel-like region called the zona pellucida. So we have the zona pellucida and cells that make up the corona radiata. They're all there, and it acts as a barrier for the sperm cells. Now, this is a good look at what a polar body is. Remember, for females, that in meiosis in females and big picture stuff, I will ask on the test, right? Differences between males and females in meiosis. That's the kind of stuff I would ask on a test. I would not ask on the test specific questions about different part of meiosis, meaning like what happens in metaphase 2 of meiosis, what happens in prophase 1 of meiosis. I wouldn't ask that specifics, but I would ask you to know the big picture of meiosis. Why is it there? Why is it different? How is it different for males than females, right? Big picture stuff. Now, if you take a look right here, what we'll see is that one of the most important things we have to do is if there's fertilization right here, if one sperm cell somehow makes it through all those barriers and now fertilizes the egg, what we need to do is make sure no other sperm cells penetrates through. So we're going to have some blocking, all right? We call it the fast and the slow blocking. And what's going to do is going to prevent other sperm cells from penetrating. You can imagine if you have one sperm cell penetrating through, that's 23 chromosomes. Immediately, mom, remember, she started, right? Basically at ovulation with meiosis 2. She never completes meiosis 2, the egg, unless there's fertilization. If there's fertilization, then the oocyte finishes meiosis 2. How do we know this? Well, here you only see one polar body, right? Now we go to number 4, because this goes in sequence. 1, 2, 3, 4, 5, 6. So number 4, you have that sperm's head here with its 23 chromosome. And now you'll see you have 2 polar bodies here. This means that this oocyte understands that it's just been fertilized and it has immediately finished off meiosis 2. So then the 23 chromosome nucleus or nuclei can fuse together to form now a zygote, a single nucleus, 23 chromosomes from dad's sperm, 23 chromosomes from mom's oocyte right here. Now they fuse, now you have 2 sets of 23.
Makes 46.
right? Exactly, right?
Yep.
Remember, it's not 46 different chromosomes. It's
Mhm.
23, but you have two sets, one from each parent. Now, when we have that early cell division, a zygote divides to form two cells at about 18 to 39 hours after fertilization. The two cells divide to form four, then eight. Now, pluripotent cells are stem cells that can basically become any other type of cell in your body. You can imagine if all we're doing is taking a cell, all right, from a fertilized
Mhm.
egg, and there's only 8 cells at that point, or God forbid, only 4 cells at that point, removing any cells, right? It's like removing a quarter of the cells in us right now. So if you form and right after fusion, if you have some kind of damage to any one of these cells when we're very early on in gestation, there's likelihood that the pregnancy will not continue. Because if you lose one cell out of 4, one cell out of 8, there's virtually, we know that one cell is going to be important. It could turn out
Mhm.
to be the brain. It could turn out to be the heart. We don't know because there's only 8 cells at that point. But we know that out of those 8 cells, every single one of those 8 will be important. All right. They can become just about anything. And we'll see with twinning, it can even become a different person. Now,
Mhm.
a zygote's the name we have it when we have 2 cells. Then when we have morula, it's a solid ball of 12 or more cells. Once we get to a point where you have a lot of cells, now we need somehow to tap into mom's blood supply. Because if we don't tap into mom's blood supply, then we're not going to have enough nutrients for us to then have any way of having more cell growth, right? Up until we get to the blastocyst phase, we have enough in mom's oocyte to keep that mitosis occurring shortly after fertilization. But you can imagine, right? If you have a small little garden, you can only feed maybe 2, 3 people. Now, what
Yeah.
happens if all your friends come out all of a sudden, and now you have 10, 20 people coming in your house? Well, that small little garden is going to do it, right? You need to go and actually get more food. You need
Mhm.
to go and drive and get more food. Same thing with the blastocyst. When you only have maybe 10 to 12 cells, you might have enough nutrients from the original mom, right? Original oocyte. But once you get to a greater number of cells, you need greater nutrients. So now we're going to somehow have to go into endometrium, and now we're going to have to eventually form a placenta and somehow tap into mom's blood supply. And this picture kind of shows us what's happening here. Usually, you're going to have fertilization in the fallopian tubes. The reason why is simple. If you have fertilization in the uterus, you don't give time for that fertilized egg to undergo many rounds of cell division, to grow, and then implant. By the
Mhm.
time you start undergoing cell division, you're basically out of the cervix and out already. So you need to have fertilization occurring in the fallopian tubes. Then you have a date that you're going to have 2 cells. Then you have multiple days. And by the time you hit about a week, what happens is you have produced so many cells. Now those cells have enzymes that are able to penetrate through mom's endometrium and implant there. So fertilization location is important because the closer we are to the ovary, the more time we have for the cells to grow, to become more competent, so then they can get into the endometrial lining and now tap into mom's blood supply. Now, what I do want you to know are what are the names of the cells that actually penetrate through, right? You have these cells right here, and it's kind of crazy. Remember, we're going to have clusters of cells, more than 20 of these cells. And what we're going to see is only a small number of them will turn into baby. Take a look, right? Look how many cells we
Mhm.
have here. And only the ones in yellow will become the baby later on. The other ones
Mhm.
right here called Sensational Trophoblast has enzymes that allows it to eat through the endometrial lining to tap into mom's blood supply deep in the myometrium. Now, I mean, that's crazy, isn't it? If you think about, we have clusters of cells, yet only this small number. And it kind of makes you realize just how amazing birth really is. Because you can, if any one of these gets damaged, right? You're not going to have a baby that's able to be birthed. And usually the baby's damaged and mom's going to have what we call a miscarriage as a result of it. Now, the Sensational Trophoblast is what are the cells that allows the placenta, that allows the baby to kind of work its way into mom's endometrium and into the myometrium. So then we can tap into mom's blood supply. As we do this, we have a second layer of cells called cytotrophoblasts that kind of makes, right, what looks like little arteries, capillaries, surrounding mom's arteries and capillaries. Now, keep in mind, there's never a time when we can have mom's blood directly go into baby, right? There's never any true mixing of blood during the normal cycle of the 10 months until labor. Now, the only time you might have mixing of blood in labor is if you will have normal vaginal delivery. Mom's bleeding, baby's coming out, there might be nick and cuts too, right? And you're going to have some kind of cell transfer. And what we're going to see then is there's a chance that there's going to be exchange of genetic material, exchange of cells. You know, right? Remember, we started talking in chapter 19 about hydrops fetalis, when mom is, right, O negative and baby is O positive, right? Or it can be A negative, B negative, but mom has negative blood, but baby has positive blood. And we talked about how there was no mixing of blood until birth, right? And at birth, then is violent. You've probably been there. Were you there through all of the babies?
Yes.
Right? Was it any of them C-sections?
No.
Yeah. And you know how violent it can be, right?
Yes,
it's not fun for the women, right? For us, we just sit there and kind of look, like amazed, right? Enjoy the baby coming out. You get to hold the baby first. Everything's great for us. For mom, though, you have violent process, and now there might be nicks and cuts because remember, baby has to go through a very small birth canal. Now, there's a belief that there's only so large that the woman's hips can become, right? So then the reason for it is simple. The wider the hips, the greater size of the brain and the head of the baby. But there's a limit to how wide we want that vaginal canal to be. So what we believe now is that we are at the 9-10 months that we're having gestation is actually much shorter, right? Usually it should be about 18-20 months, but there's no way at 20 months. You can imagine a 1-year-old that's like 30 pounds coming out of mom, right? Well, that would be the time when we would want baby to really come out and be somewhat self-sustaining, right? But that's not good for mom. So what we do is give birth to baby before they are actually supposed to, right? And when a lot of growth happens in the baby outside of the womb. In most animals, like you've seen videos of elephants, seen videos of gazelles, lions, right when they're born, they're able to walk. They're able to do things, right? But not us. And the reason is because we're born at such a young age, right? And most of the growth occurs after delivery. Now, the rest of this, right? I do want you to know what happens here. Know the name. You will see it on the test. Sensational Trophoblast, right? They're the ones that allow us to allow the placenta to go into the endometrium, to go through into the myometrium where the blood vessels are. As it forms these, the placenta right here, the blood vessels, moms, are going to be encircled by the Sensational Trophoblast in light green and the cytotrophoblast in dark green. And eventually we're going to have this kind of huge cauliflower shape to it, right? Now, what does this mean? So this was this original shape right here. And now with time, right, we made it smaller. We made it, by making loops right here, we increase the surface area. And what we're doing is bringing capillaries, forming capillaries into sinuses found in the in mom's blood. So mom's blood's going to go through these spiral arteries. As it goes through, it's going to then pool in these what looks like little caverns, right? So you have all this blood pooling in the caverns, and now you have babies, capillaries right next to it right here, right? Notice there's always cells blocking the direct interaction between mom's red blood cells in the caverns here and babies, which is stuck inside the capillaries. So there should never any mixing of blood. Now we call this, right, over here, right, the Chorionic Villus. I want you to know that name because if you're gonna have kids again, your wife is how old?
She is 38.
Okay, yeah, if you are going to have kids at the age of 30 and above, all right, most doctors are going to be worried about Down syndrome. So they will do an amniocentesis. And amniocentesis means that they're going to put an actual needle into the amniotic sac, and they're going to remove a little bit of fluid in there that should have baby cells. And then they're going to check the baby's cells and its chromosome count to see if there is a chromosomal abnormality, like trisomy 21, Down syndrome. There are worse abnormalities besides, trisomy 21. You have trisomy 18 and 13. 18, Edwards, these are genetic lethal ones. Where in Down syndrome, yeah, it's bad. They might have, right, mental deficits, things like that. But usually they can survive until they can have a nice, normal, healthy life. But if you have trisomy 13 or 18, usually they don't survive long. You might have maybe 1 in 50 that survives until teens. All right. So that's a little bit more dangerous. And the older we get, the older mom gets. It used to be we think it's only mom. The older mom gets, the greater the risk. But even with dad, the greater, the older dad gets, like us, what happens is our sperm is gonna might not have the same kind of healthiness as it did previous. Now, if they can't get any cells in the placenta and the amniotic fluid, then they'll do what's called a chorionic villus sampling. That's a little bit more dangerous because you're removing some of the cells in the chorionic villus. It can damage some of the circulation, and then it carries the risk of miscarriages. Now, I always tell my students this: if you don't care if the child is going to have Down syndrome, if you don't care if this, if you're going to love the baby no matter what, then you might not want to take the chance of an amnio or a chorionic villus sampling, right? I mean, you're going to find out after, 9 months anyways, right?
Yeah.
Now, when we talk about here, remember I mentioned in class that when we're first conceived, based on if we are XX or XY, if we're XX, we're females, and the females are going to have ovaries, right? Now, if we're XY, we're going to have males, and then we're going to have testes. Now, you're going to have testes or ovaries based on your chromosomes. Besides that, though, right, you're going to have ducts for both sexes. So early on in gestation, an XY male will have testes and 2 sets of ducts. One of the set of ducts will turn into the fallopian tube and the uterus. One of the set is going to turn into the vas deferens, is going to turn into the prostate, is going to turn into seminal vesicles. So one set of duct turns into the female reproductive system, the other turns into the males. But at conception, you have both. So we need to find out, right, wait, we have testes. Well, we want one set of ducts to continue to grow, and then another set of ducts to die off, right? So in order to understand this, we're going to figure out, all right, what are the mesonephric? What is the paramesonephric ducts? The names: Now let's take a look, and I have better ones right here. So you have two sets of ducts, right? One, the mesonephric, the other one, the paramesonephric. Now you can take a look right here. The green ones right here are the paramesonephric ducts, and it's going to turn into the fallopian tube and the uterus. The male ones are going to be called the, all right, the females are paramesonephric, the males are the mesonephric ducts. Now, some books prefer to use Wolffian and Malarian, right? I like using Malarian and Wolffian, but no both, right? Now, why do I like using Malarian and Wolffian? Well, kind of easy, because these names sound very similar, don't they? Mesonephric and paramesonephric. It can be kind of confusing. But if we use the actual, like, labeled name, right, the name actually kind of makes sense. Wolffian ducts, kind of like a wolf, right? A wolf looks kind of hairy, like a werewolf. It's going to make sense. It's going to become more of a male duct, right? Malarian ducts are going to turn into female ducts, right? So if you understand one and know the name of one, you should know the name of the other one. So if you know one set of ducts and turn into the Wolffian ducts, and then the vas deferens, well, wolves are hairy. And because wolves are hairy, guys are usually pretty hairy too. So we're more likely to have the Wolffian ducts, right? Just a silly way of just hopefully help you kind of understand which one is which. Remember, Wolffian ducts, mesonephric. Malarian ducts, paramesonephric. Both are present, even though you have either XY and have testes, or you have XX and have ovaries, right? Now, this is good because, we can tell, right? Eventually, if we have ovaries, we're going to have female ducts. We have testes, we're going to have the male ducts. The problem becomes if there's an issue in utero. You guys have probably, and you've probably heard of having people born with indeterminate gonads, right? Where we don't know if it's a boy or girl. Some people use that term hermaphrodite. We don't use that term anymore. But, back in the old days, you've heard horror stories on TV shows about how mom or dad, the baby's just born. We don't know what's going on, right? It has testes, but yet we know it should be a baby, or it should be a girl, right? And it should with scrotum that's hanging out. So what do? Right? Most of the time they want to do it pretty quick after birth. So then baby is going to be healthy afterwards, right? The problem is you make that decision so early on that you can cause symptoms and you can cause some mental health issues. But the reason why you're able to have that is because you have both sets of ducts at gestation, right? Now, what happens? How do we get one set of ducts to get bigger and stronger and to mobilize? Well, the second pair of ducts start to die off and undergo what we call atresia. Now, this is pretty cool. There are 2 times, all right, when we will have the release of testosterone. The first time is at puberty. Makes sense, right? The second time is really early on in gestation. So what happens? Since males have the Y chromosome, there's a specific gene called the SRY gene, right? It's a sex determining region, right? So this gene is on the Y chromosome only. This gene activates in utero. In the process of activating, it activates the Sertoli cells. Remember, they have like 10 different names. The Sertoli cells, the sustenticular cells. Those cells right there will secrete Malarian inhibiting substance, which then causes apoptosis of the Malarian ducts. That's why I want you to know it as both Malarian and mesonephric, paramesonephric, right? So what do I want you to know? Right. So in, basically the start of the second trimester, end of the first, this gene on the Y chromosome, SRY gene, is going to activate. It's going to cause Sertoli cells to release Malarian inhibiting substance, causing the female paramesonephric Malarian ducts to die off. Then the primitive gonad tissue, right, is going to start to release testosterone. Testosterone causes the male Wolffian ducts to develop. As it develops, it's going to turn into the epididymis, the vas deferens, the ejaculatory duct, all the other stuff that we kind of determine that's required for male sexual reproduction. All right. So again, it is the male Y chromosome that actually starts this whole process. Now, what happens if you're a female and you're XX? In the female, you don't have that Y chromosome, so you don't secrete the Malarian duct. You don't have testes, so you don't secrete testosterone. So what happens for females, female ducts, the female Malarian ducts will develop as a default, right? It's a default in the absence of Malarian inhibiting substance and testosterone. So when nothing happens, if there's no testosterone release, right? So you can even have an XY boy, right? But for some reason, this gene is mutated. Well, what happens if it's mutated? It can't secrete Malarian inhibiting substance. When that happens, the female Malarian ducts don't die off, right? If the chromosome isn't, functional, you also won't release testosterone. So the male ducts don't develop. When they don't actively get developed, they die off. So the Wolffian ducts in that XY male with, a mutation now, well, the male ducts start to die off. And the female ducts is the default. And you're no longer releasing Malarian inhibiting substance. Because of that, the default ducts start to develop here. This is where we can get, I don't know if you've seen the picture I have here, right? These are all XY males. Every single one of these are XY males. Crazy, right? They have androgen insensitivity at a time when testosterone should have been released in utero, where Malarian inhibiting substance should have been released in utero, right? Even though they have, the XY chromosomes, right? It didn't get released. So what happens? The female genitalia will develop. And instead of connected with the ovaries, they're going to be connected with very, very small underdeveloped testes in the hip. Crazy, right?
Yeah.
Right. You can imagine the worst part about this is they will develop like girls because they have completely female genitalia. They will develop like girls. Nothing, they would probably not even know that they're anything but girls until they hit puberty. And when most girls will start having menses and start having their periods, well, they don't have ovaries, right? So they're not going to have menses to the same extent, right? And that's when we find out that, wait, there's something wrong here, right? Because usually we don't do, and there's a black and white picture, so it's old, right? Back then, doing genetic testing is super expensive. Even now, it can be super expensive. Now, again, right, in the female, no SRY because there's no Y chromosome, then the female ducts develop. In males, testosterone is released, gets converted to DHT as well, causes the genital tubercle to develop into urethra, prostate, and the genitalia. In other words, testosterone is a positive driving agent. It causes all the male system to develop. Kind of crazy, but we don't see this. It's pretty rare. So every time, if you go on YouTube, you go on do some bad Google searches, you would think that it's common. It's not very common, right? So kind of review this. I'm not, again, I go into a lot more detail, kind of discuss the cells that are activated. Again, it's nothing, we hadn't discussed just a few slides ago. Now we're just adding the name of the cells here. Sertoli cells secrete Malarian inhibiting substance, causing the female Malarian ducts to undergo atresia and die off. Then Leydig cells secrete testosterone, right? Remember, the Leydig cells are in the testes, they secrete testosterone, causing the male Wolffian mesonephric ducts to develop. Testosterone then stops secretion at birth. All right. So you have the secretion in utero, and then it stops until you hit puberty. Kind of crazy, but there's an off switch. Now, right. Again, kind of review this and the genitalia stuff that we kind of mentioned. Again, with androgen insensitivity, usually these people, when there's ever an issue where there's testes and then what looks like fallopian tubes and uterus, these, all right, all three of them are sterile, right? So whenever there's usually an issue with the gonads, so the ovaries or the testes, or where there's an issue with the urethra, ureter, right? That kind of stuff, right? Anything with the sexual reproductive system. If there's any kind of mismatch, it's almost automatic that all three of them will be sterile. They won't be able to have any kids. Now, if they have testes, it's sitting into the abdomen for so long that they won't have any viable sperm either. So keep that in mind, that, it's kind of a make sure thing. So then we don't have, these males trying to carry a baby, which is awkward, right? All right, now, the last thing we're going to talk about are just some of the changes that we see. So some of the changes with parturition or labor and delivery, and what happens in the fetus. Some of this we talked about. Remember, we talked about the foramen ovale, right? Remember, we talked about the Ductus Arteriosus that becomes a ligamentum arteriosus. So what we're going to see is that I'm not going to ask questions about labor and delivery. I will ask questions about some of the hormones that allows us to have labor and delivery. And the biggest one and the drop in the hormone levels is progesterone levels, right? At birth, progesterone levels start to level off and starts to dip. Remember, progesterone is the hormone of pregnancy. If for any reason, at any time, progesterone levels start to drop off, you will have an immediate shedding of that placenta and the baby. So if you're in the second trimester and for some reason the progesterone levels start to die off, right? It starts to really go down. Immediately, that's a trigger for labor and delivery. At about the mid-second trimester, right? At about 6 months, end of the second trimester, that's about the edge of viability. If baby's born at that age, 6 months, maybe a week earlier, we have amazing meds, amazing ICUs and NICUs that can help the baby grow and develop outside of the womb. But before that, 23 weeks, right? Babies usually don't survive without any kind of deficit, meaning that they might survive, but they might have some mental deficits, or they might have lung problems, or might have GI issues, right? They might survive, but there's something that's going to be wrong, right? Now, the biggest driver of parturition or labor is progesterone. The reason why I'm mentioning it is because we did mention it in chapter 28 as well. Now, you have probably heard of the abortion pill. The abortion pill is not the actual morning after pill. The morning after pill is going to be high in estrogen. And what the morning after pill does is that you think, you have unprotected sex that night before, and now you might, be pregnant, right? So if you don't want to have a baby and don't want to get, like a dilation and curettage, like a real abortion, you might take the morning after pill. The morning after pill, that high level of estrogen causes an immediate shedding of the endometrium. So now the endometrium shed, even if you have a fertilized egg coming down the fallopian tube, you're not going to have any place for that egg to implant because the endometrial lining is gone, right? That's a morning after pill. Now, the morning after pill is not useful if you're already pregnant, though. If the baby has already gotten its placenta in here, it has already implanted. That high estrogen is not enough to cause shedding of the entire endometrium and the pretty large forming fetus. So the actual pill that is the abortion pill blocks progesterone completely, right? And by blocking progesterone, you do cause the whole endometrial lining to be shed. But again, though, if you have a baby that big, right, and you shed the lining, what do we call that? We call that giving birth, right? So yeah, that's that's why we want to do it before the end of the first trimester, right? Now, I'm just discussing it as a medical procedure, right? My parents are religious; they would even kill me if I even discuss it, right? And probably get mad at me discussing it in class. So just understand, I'm not advocating it. I'm not advocating it either. So just understand we're just talking about it because there are things that you will hear about in your professional life. And I want you to know what the basis of it are, okay? Now, what things happen in the newborn? And I'm gonna highlight the ones I want you to know. In utero, there's no blood going through the lungs. We kind of talked about it. Why? Because there's no air in the lungs, right? If there's no air in the lungs, there's no reason to bring blood there because all they will have is carbon dioxide. So what happens? Well, we're going to redirect and detour blood from the right atrium to the left atrium by way of the foramen ovale. So remember, just a little hint, right? You have the right atrium, blood goes to the right ventricle, then it goes up the pulmonary trunk into the pulmonary arteries, into the lungs, goes back through the pulmonary veins, through the left atrium, through the left ventricle, up through the aorta, through your systemic circulation. Now, we don't want blood to go into the lungs because there's no oxygen there. If you do bring blood in the lungs, you'll just pick up more carbon dioxide, which is something that can be bad for us. So we're gonna detour blood away from the lungs. So we're gonna have two shunts. First shunt, foramen ovale. What does it do? It allows blood to go from, in utero, that foramen ovale was the fossa ovalis, right? And what happens? It allows blood to go from the right atrium to the left atrium. What have we done? Detoured some of the blood that's in the right side of the heart. Now we bring it to the left side. While the blood that goes to the left atrium, we've done its job, right? We've detoured it from the lungs. Instead of going to the lungs, right, blood that goes into the left atrium bypass the lung. Now, not all of the blood, though, can go through that fossa ovalis, right?
Yeah.
Now, the second shunt right here, and there's three shunts, right, is going to be called the Ductus the Ligamentum Arteriosus, right? Well, that's the Ligamentum Arteriosus when we kind of discussed it in class, right? In utero, it's a Ductus Arteriosus. I know it's kind of confusing. When I write a test question, I will actually ask, I'll be definitive, right?
All right.
I'll be like, all right, this, in our class, when we discussed the cardiovascular system, the Ligamentum Arteriosus was open, and it was called the Ductus Arteriosus in utero. What happens, right? What happens is some of the blood that goes from the right atrium goes through the foramen ovale, the fossa ovalis here, and into the left atrium. But that's a pretty small hole in the heart. So not all of the blood from the right side can go through that small hole to the left atrium. So a lot of the blood then is going to go from the right atrium to the right ventricle. As it goes up the right ventricle, it goes up the pulmonary trunk. As it goes up the pulmonary trunk, it goes through the ductus arteriosus to the aorta. So this is a shunt that connects the pulmonary trunk to the aorta, allowing you to, again, bypass the lungs. All right, so these are the first two shunts I want you to know. The last shunt is going to be found in the liver right here, right? The ductus venosus right here. The ductus venosus right here, all right? Remember, we should have the hepatic portal vein bringing nutrients into the liver. Nutrients we've just eaten, right? We've just eaten, we've digested it, we've absorbed it. It makes sense that we're going to bring all that nutrient-rich blood to the hepatic portal vein so then we can then bring it to the liver. So then as we bring it to the liver, guess what? Right? The liver gets first dibs out of the nutrients. It can produce glycogen for the body and the brain to use. It can produce amino acids. It can produce fats. Now, in utero, are they eating? No,
Mm-mm.
right? They're receiving mom's blood, right, at the chorionic villus. So what's happening in utero is there's no nutrients going into the GI tract. So why bring that extra blood into the liver when it really doesn't need it? Right? So what happens? You'll have some blood supply going into the liver. The vast majority of the blood supply goes directly into the abdominal, to the inferior vena cava, bypassing the liver. Why? Well, we don't really need the liver to be functional in utero because, right, you're receiving all of the nutrients from mom's storing all the nutrients for the baby, and more importantly, mom's getting rid of a lot of the waste.
Mm-hmm.
Now, what happens though, right? This is a reason why newborns, even full-term newborns, can be somewhat jaundiced. In full-term newborns, even full-term ones that are, maybe full 3 trimesters, then you give birth at 10 months. Well, what
Mm-hmm.
happens? Well, even then, the liver never had to work. Because the liver never has had to really work in utero. When baby is born, it takes a while for the liver to actually mature and then to activate and do its job, because it's never had to before. And in the process of waiting for the liver to mature and do its job, you're going to be producing a lot of bilirubin. Remember,
Mm-hmm.
bilirubin was produced by most usually in the spleen, which they don't show here. So in the
Mm-hmm.
spleen, we recycle 2 million red blood cells every second. In the bone marrow, we make 2 million red blood cells every second. In the spleen, we get rid of 2 million. In the process of getting rid of 2 million red blood cells, the globin chains of hemoglobin are proteins. So we can reuse the amino acids that the proteins are made out of, right? Easy. We reuse all the globin chains from hemoglobin. The iron we reuse really easily. The heme turns into bilirubin, right? So the more you have red blood cell destruction, the more bilirubin you have. But keep in mind, in utero, mom was getting rid of all the bilirubin. Now there's no more mom because you're just a 1-day-old baby. What happens? The liver has never had to work before, or if it did, very, very minimal activity. Now it has to do all the job that mom was doing before. It had to take all that bilirubin because you're still making 2 million red blood cells a second. You're still recycling 2 million red blood cells in the spleen every second. Now you don't have mom to remove it. So now the liver has to do a lot of its job immediately. And at first it can't. And what happens? The bilirubin starts to build up and starts to be deposited, right? At first it deposits on your skin and your, subcutaneous tissue, and then the sclera of your eyes and even in the mucous membranes. Now, if the bilirubin levels get too high, it can cause permanent mental deficits. So it can cause, right, severe mental issues. Nowadays, we're a little bit more aggressive in the treatment, and we give the baby, the newly born baby, Billy lights and Billy blankets. And the lights allows you to disperse the bilirubin, decrease in the concentration. All right. That's all I want you to know.
All right.
All right. Now with APGAR, yeah, it's a little bit, you guys will probably talk more about it later on, but that's it.
Okay.
Do you have any questions?